Today we'll be talking about amino acids. Amino acids are important to us on two different levels. At the first physiochemical level, these amino acids act as important buffers in the body. An amino acid is made up of an amine group, which is a weak base, and an acid group, which is a weak acid. So they can act as a buffer over a wide range of pH, from an acidic range all the way up to the basic range. On a structural level, amino acids make up several proteins which have several different functions in our body, and that's the reason we are studying amino acids. These are the objectives. So, a protein is a vital molecule that carries out several different dynamic and structural functions in the body. Proteins are all similar in that they are made up of chains of amino acids. These basic building blocks of proteins are linked together by unique chemical bonds called peptide bonds. So you could take a lock of your hair or a pile of scrambled eggs, and even though they look very different, at their molecular core, they are simply chains of amino acids bonded together. So what are some of the dynamic functions of proteins? Number one is it's used to transport materials, an example of which is transport of oxygen by hemoglobin. Hemoglobin, which is the oxygen transporting protein, is found in your red blood cells. So when you breathe air into the lungs, the oxygen molecules bind to the protein hemoglobin of a red blood cell so that it can travel anywhere in your body. Proteins play a role in metabolic control by hormones. There are several body processes which are influenced by hormones. Hormones are proteins that regulate the activity of cells or organs. They are like chemical messengers that can carry an order from one part of your body to another. For example, a common hormone is insulin that regulates blood sugar by carrying a message to your body cells about how much sugar is present in your blood. Proteins play a role in contraction of the muscles in your body. With the help of proteins such as actin and myosin, your muscles can contract and relax. Next, proteins regulate body processes. For example, enzymes are nothing but proteins that speed up chemical reactions in the body. Without them, basic activities like breaking down the food you eat would happen too slowly to support your metabolism. You can think of enzymes that are as proteins that shift your body processes into the higher gear. Next, they can help with immunity. For example, antibodies are nothing but proteins that can recognize foreign invaders in your body, and these vigilant proteins work together with other cells from your immune system to help prevent infection, illness, and disease. The most important structural function of protein includes providing matrix for bone and connective tissue, which eventually gives structure and form to your body. An example of which is keratin, which is found in your hair, nails, and skin, that gives these specific tissues a specific structure. Another example is collagen, which is a structural protein that is found in various connective tissues. It provides the framework for the ligaments that hold together your bones and tendons that attach the muscles to those bones. What are the specific functions of amino acids? Amino acids are involved in the biosynthesis of proteins, enzymes, and hormones. Okay, to form a protein, amino acids are linked by dehydration synthesis to form peptide bonds. The chain of amino acids is also known as polypeptide. Some proteins contain only one polypeptide chain, while others, such as hemoglobin, contain several polypeptide chains twisted together. Then amino acids also play a role in biosynthesis of nitrogen containing biomolecules. You will hear more about this when you talk about nucleotide metabolism. But in a very brief manner, what this means is there are several amino acids such as glutamine and aspartic acid that donate their nitrogen and carbon atoms in the synthesis of purines, adenine and guanine, and pyrimidines, which are cytosine and thymine. And then some amino acids can be metabolized to produce energy. We did talk about this a little bit when we talked about the chapter nutrition. So how this works is dietary proteins are first broken down to individual amino acids by various enzymes and hydrochloric acid present in the GI tract. These amino acids are further broken down to alpha keto acids, which can be recycled in the body for generation of energy and production of glucose or fat or other amino acids. The breakdown of amino acids to alpha keto acids occurs in the liver. So, structure of an amino acid. Amino acid has four important parts. An amino acid has the amine group, amino group, it has the carboxyl group, it has the alpha carbon, which is the carbon between the carboxyl and amino groups, and it has a distinctive side chain. Now, these four moieties remain the same. Okay, what makes an amino acid distinct is this distinctive side chain, and there are a total of 20 amino acids. I did mention that amino acid can act as a buffer over a wide range of pH. That is because you have the pKa of carboxylic group, which is approximately between 1.8 to 2.4, and then you have the pKa of amino group, which is between 9.1 and 10.8. So because of these two varied pKa values, amino acids can act as a buffer over a wide range of pH. These amino acids are combined to form a polypeptide chain through the help of peptide bonds. Okay, you have the carboxylic group of one amino acid coming together with the amino group of the second amino acid, losing a water molecule through a process of dehydration to form this peptide bond. So this is how exactly a peptide bond is formed. Here you have the hydroxy group of the carboxylic acid. Here you have the hydrogen coming from the amino group. Together through the process called dehydration synthesis, it loses a water molecule, and you have the carbon atom of the first amino acid forming a peptide bond with the nitrogen atom of the second amino acid. This is what a peptide bond is all about. So the process is called dehydration synthesis that results in a loss of water molecule. The amino group loses a hydrogen, the carboxyl group loses the hydroxy group, and eventually you have the nitrogen of the amine group bonding with the carbon of another amino acid's carboxyl group. So what is Fischer projection? Fischer projection is nothing but a method of a three-dimensional representation on paper. Okay, so amino acids are three-dimensional moieties, and they can exist in two different forms. One of them is called the L amino acid, the other one is called the D amino acid. L amino acid is the one that is synthesized in the cells in humans. Human proteins do not have D amino acid. They cannot incorporate the D amino acid. Some plants and some bacteria can incorporate the D amino acid, but not humans. So coming back to Fischer projections, what it really means is these solid arrows, the one between the amino group and carbon and the one between the carbon and hydrogen. The solid arrows, if you have to look at this and imagine, you can imagine them as coming out of the plane of your computer screen. The dotted groups, the one with the amino acid side chain and the carboxylic acid group, are going inside and away from you towards away from the screen. So that is essentially what a Fischer projection is all about. So using a Fischer projection, you can represent the L amino acid, you can represent the D amino acid. How do you remember which form is the L, which is the D? The easy way to do that is this is an amino acid. So it's all about the amino group. So whenever you have the amino group facing left or towards your left hand, that is the L amino acid. The D amino acid has the amino group on the right hand side. Chirality. Chirality is a very important concept in chemistry. The concept that some things are identical to their mirror images and others are not is a fundamental principle in organic chemistry. The adjective chiral is used to describe molecules that are not identical twins with their mirror images. A chiral molecule cannot be superimposed on its mirror image no matter how you rotate it. You can say that your body is chiral. An achiral image molecule, on the other hand, cannot be distinguished from its mirror image. Now, as an example, you have this person here who is wearing a watch on his left hand. The mirror image has the watch on his right hand. Now, there's no way if I were to put them side by side, so this is the mirror image, this is the actual person wearing his watch on his left hand. There is no way, no matter how you rotate this, that you can superimpose this image on its mirror image. That's why these two molecules are chiral. On the other hand, you have when you have a superimposable mirror image, like the chair in this picture, it is considered to be achiral. Now, if I were to put the chair and its reflection side by side, all I have to do is just move one on top of the other, and I have a superimposable mirror image. That is called achiral. So the reason we talked about chirality is because amino acids are chiral molecules. Instead of looking at every molecule in a mirror and rotating it to say if it's chiral, how else can you tell if a molecule has this property? And the answer is pretty simple. A carbon atom that contains four different substituent groups will be chiral. That particular atom is called a chiral center. When two of the groups on a carbon atom are the same, that carbon is not a chiral center and the molecule is achiral. One such amino acid that is achiral is glycine. Glycine has two of the same substituents, which is the hydrogen atom that makes it achiral. Now, what are enantiomers? Enantiomer is just a slightly shorter way of saying non-superimposable mirror image. All chiral molecules have enantiomers, which are unique molecules in their own right. An achiral molecule doesn't have an enantiomer. Its mirror image has the exact same identity as itself. So, why do you, as a pharmacist, need to care about these chiral molecules? Well, it turns out that nearly every molecule in the human body or the biological systems is chiral. Often, only one enantiomer, that is, either the molecule or its mirror image, works properly to carry out a given biological function. Now, this is important to consider when you're designing pharmaceutical drugs. An example of which is the active ingredient in the sleep aid Lunesta, which is a chiral molecule. Though this molecule helps people with insomnia, the mirror image of this molecule doesn't do anything. In other cases, the mirror image of a medicine's active ingredient could actually be harmful. The thalidomide disaster is one of the darkest episodes in pharmaceutical research. This drug, which was marketed as a mild sleeping pill, safe even for pregnant women, actually turned out that it was the one of the ingredients, the other enantiomer of thalidomide, was actually tetragenic. So as a result of which, thousands of babies worldwide were born with malformed limbs. Classification of amino acids. Amino acids are classified based on their side chains because it is the side chain which gives the amino acids its distinct property. Now, all of these side chains, all of these amino acids that you're looking at on this slide have non-polar side chain. Now, from the first lecture, if you can recollect what non-polar is, it is really so. When we talked about hydrocarbons, all of them are non-polar. The bond between carbon and hydrogen is a non-polar bond, as the electronegativity difference is probably around zero point 0.4, which is not that big of a difference. So all of these amino acids with side chains that have hydrocarbons or even a hydrogen are non-polar. So you have glycine, alanine, valine, leucine, isoleucine, phenylalanine, all of which have hydrocarbons. All of these are non-polar. Then you have tryptophan, which in addition to the hydrocarbon has a nitrogen. Again, the electronegativity difference between carbon and nitrogen is insignificant. To figure out exactly how much that difference is, let's move on to periodic table. So you have carbon, which is 2.5, nitrogen sitting right next to it is 3. It's not that big of a difference, again making this bond non-polar. Likewise with methionine, you have a carbon that's bound to sulfur. Well, they actually turn out that they have the same electronegativity, so there is not any difference in the electronegativity, making again methionine, which has a sulfur, a non-polar side chain. And finally you have proline, which is again a hydrocarbon. Now, one thing to note in all of these amino acids depicted in your textbook is that your amino acids are always shown to be in protonated form. What that means is wherever your amino acid can gain a proton, it has been shown to have gained a proton and therefore it is in protonated form. So you have two places where an amino acid can get protonated. One is the amine group, the other is the carboxylic acid group. Both these groups have been shown to be protonated. An amino acid does not always exist in its protonated form. We'll talk about this a little later. The next group of amino acids are the uncharged polar side chain. So what that means is the molecule on a whole is uncharged, carrying a net charge of 0, but the side chains are polar. And it's kind of easy to imagine why. So if you again go back to the first lecture, the hydroxyl bond in water is a polar bond, and that's exactly what makes some of these amino acids polar. They have a hydroxyl bond. So oxygen is highly electronegative, kind of tends to pull the electron towards itself. That gives the hydroxyl bond its polarity. So all of the amino acids that contain the hydroxyl bond, serine, threonine, tyrosine, are polar. And then you have asparagine and glutamine, that while they do not have the hydroxyl bond, they have carbon that is double bonded to oxygen. Okay. So the electronegativity difference is fairly high. Oxygen is on the increasing electronegativity side. So that makes the carbon-oxygen bond polar, which gives these two side chains, asparagine and glutamine, their polar nature. That brings us to the last one in this group, which is cysteine. Cysteine contains the thiol group. It contains the sulfur attached to hydrogen, which is considered to be a thiol group. Now, cysteine as an amino acid and its polarity falls somewhere in the gray area. It's one of those amino acids that's considered to be sitting on the fence because the sulfur hydrogen bond is considered to be very, weakly polar. Okay, the electronegativity difference is not as much. It's about zero point 0.4, but sulfur is considered to be slightly electronegative, which gives the thiol bond its weak polarity. There are several textbooks that list cysteine as being non-polar, and then there are some textbooks that list cysteine as being polar. And then the next group is amino acids that have acidic side chains. And you can see that a part of the side chain of aspartic acid and glutamic acid is the carboxylic acid group. So that makes these amino acids have two carboxylic acid groups. They have one carboxylic acid as a part of their backbone structure, the core structure, and then they have another carboxylic acid group as a part of their side chain. So these amino acids now have three buffering ranges. They have three pKa values. They have two pKa values along the amino group and the carboxylic group of the core, and then the third, or rather the second carboxylic acid group has a pK value of its own. Likewise, we have basic side chains. It's easy to imagine that basic side chains, just opposite of the acidic side chains, have an additional amino group. So histidine, lysine, arginine, all of these amino acids have the additional amino group in addition to the existing one in its backbone. So just like the acidic side chains, basic side chains therefore have three pK values. Two pK values for the amino group and the carboxylic group in the core, and a third pK value for the amino group in the side chain. So amino acids oftentimes use abbreviations. Okay, you have amino acids that have 3-letter codes and amino acids that have 1-letter code. The 3-letter codes are oftentimes based on how they sound. So cysteine has cis, histidine has his, isoleucine has ile, methionine met, serine acer, valine val. And a lot of these amino acids have their first letter code, or rather 1-letter code, as their first letter. Then you have most commonly occurring amino acids with priority. So alanine is ALA, gets the 1-letter code as A. Glycine has GLY, leucine, Leu, proline, Pro, threonine, THR. And then you have some similar sounding names. Arginine is ARG, and the 1-letter code, since A is already used up by alanine, has to be R, because the R pronunciation in arginine is quite heavy. Then you have asparagine, which is abbreviated as ASN, and because again the A is taken up, it has a 1-letter code as N. Aspartate, ASP, because it has a D sound in it, it has a 1-letter code which is D. Glutamate, GLU, so the glut E is more pronounced, giving the 1-letter code to glutamate as E. Then you have glutamine. It kind of sounds like cutamine, giving it the 1-letter code Q. Phenylalanine has PHE, and phenyl sounds like F, giving it the 1-letter code F. Tyrosine is TYR. Ti, the gets it Y, since T is already used up by threonine. And then you have tryptophan. Tryptophan is the only amino acid that has a double ring, abbreviated as TRP, with a 1-letter code of W. So how are the amino acids located in these proteins? When these amino acids come together to form proteins, how do they position themselves on the polypeptide chain? So proteins can be either soluble or they can be hydrophobic or lipid soluble. So mostly you will find these hydrophobic or lipid soluble proteins in the cell membrane, because cell membrane by itself is made up of lipids. So if you were to have a soluble protein, as the protein folds into a three-dimensional shape, you can imagine, you can close your fist and imagine that as the protein being folding, you will have all of the um, polar amino acids cluster on the surface of the folded protein. The reason being, the folded protein is in contact with water, and the polar amino acids are the hydrophilic ones that want to be in contact with water. All of the non-polar amino acids are going to cluster at the center of the folded protein. They are going to try to get away from all of the water. In the opposite manner, for a membrane protein, you have all of the non-polar amino acids cluster on the surface of the membrane proteins, because these non-polar amino acids want to get away from the water and they want to be closer to the other lipids. So all of these non-polar amino acids of a membrane protein will line up on the surface in the part of the cell membrane, and then on the extracellular side or in the intracellular side in the cytoplasm, you will have most of the polar amino acids line up on the surface where they come in contact with water. So that is how the polar amino acids and non-polar amino acids position themselves on the surface of a folded protein. Let's talk about the essential and non-essential amino acids. So there are 10 amino acids which are essential, and there are 10 other amino acids which are considered to be non-essential. The essential amino acids are the ones that you must get through the foods you eat because your body can't synthesize them. So 10 out of the 20 amino acids are essential, but adults only need about 9 of them. The 10th amino acid, histidine, is only essential for infants. Your body doesn't store amino acids, so it needs a regular daily supply of the essential building blocks. Now, how do you remember which of the amino acids are essential and which ones are non-essential? You can remember this using a mnemonic. So let's call the essential amino acids private team hall. Okay, so the essential amino acids which have to be obtained from food sources include phenylalanine, valine, threonine, tryptophan, isoleucine, methionine, histidine, arginine, leucine, and lysine. Now remember that this is just a mnemonic that's made to make remembering the essential amino acids essential. So all of these are not one letter codes, especially this one right here. That's arginine. It should have been a R, but it's a just to make our life easier. So if you use this mnemonic to memorize which of these amino acids are essential, do remember that these are not one letter codes. Non-essential amino acids. The word non-essential is a little misleading because these amino acids actually play very essential roles, but since they are synthesized by the body, they are not an essential part of the diet. Of the 10 non-essential amino acids, 7 are called conditional amino acids. These conditional amino acids, if you are sick or under significant stress, your body may not be able to produce enough of these amino acids to meet your needs. The list of conditional amino acids includes everything other than the truly non-essential amino acids, which are alanine, asparagine, aspartate. So your body can make these 3 amino acids at all times of sickness and health. The other remaining 7 amino acids are conditional. Let's try to understand the concept of ionization of an amino acid. Now, an amino acid has two important functional groups. One is the carboxylic acid group, which is a weak acid and can be a proton donor. So, in its ionized form, you end up getting the anion with the proton that can be donated. The amino group is a weak base, which can be a proton acceptor. So this weak base, upon accepting a proton, can become ionized. So it's protonated and ionized. Remember, the weak acid is protonated and unionized. It becomes ionized when it donates the proton. So now with that information, let us also review something that a lot of us had some difficulty with. If you remember this slide from the very first lecture, this slide applies for a weak acid or a weak base. Now when the pH is equal to the pKa, you always have equilibrium. You're going to have equal amounts of protonated and deprotonated forms for a weak acid or a weak base. When the pH becomes less than the pKa, the protonated forms predominate. When the pH is higher than the pKa, the deprotonated forms predominate. Now let's see how that applies to amino acid. So here you have an amino acid which is alanine. The side chain on alanine is this methyl group. Okay, so the pKa of the carboxylic acid group is 2.3. Here you have put alanine in a solution that has a pH less than 2. So let's see really what's happening with both of these amino groups and the carboxylic acid group. So if you were to consider the amino group, the pKa of amino acid is 9.1. The pH is less than 2. So for this particular group, the pH is less than pKa. Therefore, it is going to be in its protonated form. For the carboxylic acid group, the pKa is 2.3. The pH of the medium is less than 2. It's slightly less than the pKa, but nevertheless it is less. So since the pH is less than pKa, the carboxylic acid group is going to be protonated. Now what's really happening with this molecule is you have carboxylic acid group which is now neutral, and you have the amino group which is carrying a +1 charge. Your weak base is not always in this form, it is in this form, but here now it has become protonated. So now it's carrying a net charge of +1. Your weak acid is always going to be in this form. That is what makes it a weak acid. So even though it's protonated, the net charge on this is 0, and because the weak base is protonated, the net charge on this is +1. Now let's see what happens when you move this alanine to a solution that has a pH of approximately 6. Now really what's happening with the carboxylic group here is, now the pH is greater than the pKa of carboxylic group. So when pH is greater than pKa, the deprotonated form is going to predominate. So here you have your carboxylic acid group losing a proton and becoming deprotonated and ionized. What's going on with the amino group? The pKa of amino group is 9.1. The pH of the solution that you put your alanine is 6. So the pH is still less than the pKa for the amino group. The amino group therefore remains protonated. Now the net charge on amino acid at this particular point is you have a +1 on the amino group, you have a -1 on the carboxylic group. So you have a +1 cancelling the -1, giving your alanine a net charge of 0. Now let's move this alanine to a further basic solution where the pH is greater than 10. Now let's see what's happening with the carboxylic acid group. The pH is greater than pKa of carboxylic acid, so it is going to be deprotonated. The pKa of amino group is 9.1. The pH of the solution is 10. So again, the pH is greater than pKa of the amino group. So now it is going to get deprotonated. Now remember, a weak base getting deprotonated does not make it ionized. Now it is NH2. So now you have the amino group which is neutral, and then you have the carboxylic group which is carrying a -1 charge. So now the net charge on this molecule is -1. That is how, if you move an amino acid over a wide pH range, the charges on the amino group and the carboxylic group can go on changing. Your amino group and carboxylic acid group can either become protonated and deprotonated depending on the pH, changing the net charge on the amino acid. Which brings us to what is a zwitterion. A zwitterion is amino acid with zero net charge. The amino acid has individual positive charge and individual negative charge, giving the net charge on the molecule 0. So here your molecule at pH 6 is a zwitterion. The isoelectric point is the pH at which the amino acid is in zwitterionic form. The way you calculate the isoelectric point for any amino acid is you average out the two pK values. So if you were to average out the pK values of 2.3 and 9.1, it would be somewhere close to pH 6. So typically your isoelectric point is the pH at which the amino acid is in zwitterionic form. So now that we know how exactly the amino acid goes through various ionization stages, let us take a look at the titration curve of alanine. So what we are really looking at is how the charges on alanine change over a continuum of pH. In the previous slide, when we talked about the ionization, we specifically took a look at three different pH points: less than 2, pH of 6, and pH of greater than 10. But if you really were to move alanine over a pH range of 1 to say 14, how would the charges on alanine actually change? That is what is called the titration curve of alanine. You are actually going to draw a curve. So let's see what's going on. At a very acidic pH, let's say at a pH of 1, the pK of carboxylic acid group is 2.3. The pK of amino group is 9.1. So the pH is less than pKa of both the groups, which means both these groups, because the pH is less than pKa, both these groups are going to be protonated. Okay, so the net charge on the molecule at this point at a pH of 1 is going to be +1. Now as you go on increasing the pH, you will come across a point where the pH is equal to 2.3. Now 2.3 is also the pKa of the carboxylic acid group. So remember again from the previous slide, when pH is equal to pKa, there is an equilibrium between the protonated and deprotonated form. Okay, so at a point where the pH is exactly equal to 2.3, your protonated form, your form 1, and your deprotonated form, where the carboxylic acid group is getting deprotonated, your form 2, are going to be in equilibrium, which means you're going to have 50% of your molecules of alanine in form 1, you're going to have 50% of your molecules of alanine in form 2. Now as you go on further increasing the pH, the equilibrium is going to shift more and more towards form 2, where there will come a point at a pH of 5.7 where all of the molecules of alanine will exist in form 2. The reason being, now the molecules in form 2 have a net charge of 0. They exist in zwitterionic form. So 100% of your alanine molecules exist in zwitterionic form, and that is what a pH, rather a PI or isoelectric point of an amino acid is all about. It is that pH at which all of the molecules of amino acid carry a net charge of 0. Now you further go on increasing the pH. Now your equilibrium starts moving towards your third stage, which is where both your amino and carboxylic acid groups are deprotonated. So as you go towards the point where pK is equal to 9.1, or rather, so the pH is equal to 9.1, which is the pKa of the amino group, at this point you're going to have an equilibrium between form 2 and form 3, because at pH is equal to pKa, your half of your amino groups are going to be protonated, half of them are going to be deprotonated. Remember, at pH is equal to pKa, equilibrium exists between the protonated form and the deprotonated form. So now as you go on further increasing your pH, the equilibrium will shift more and more towards form 3, where there will come a point at a pH of about, let's say, 10 or 11, all of your alanine molecules will carry a net charge of -1. That is what a titration curve of an amino acid is all about. Let's take a look at another example of a titration curve of an amino acid. The amino acid in question here is glutamic acid. Now remember, glutamic acid, or any amino acid that is acidic or basic, carries an additional carboxylic acid or an additional amino group. So this is the structure of glutamic acid, where you have your backbone, where you have the carboxylic acid and the amino group attached to the alpha carbon, and then you have an additional carboxylic acid group as a part of the side chain. So now this amino acid is going to have three pK values, one for each of the uh, functional groups. So the pKa of the carboxylic acid group that lies along the backbone of the amino acid is 2.2. Now let us assume that our glutamic acid is at a pH of 1. At a pH of 1, your glutamic acid is going to exist in this form because the pH is less than pKa. So everything that can be protonated is going to be protonated. Your carboxylic acid groups are protonated, your amino group is protonated. Now let's move along the pH scale. Now what is going to happen exactly at the pKa of 2.2 or a pH of 2.2 is, because the pH is equal to pKa, exactly half of the carboxylic acid groups are going to be protonated and the other half are going to be deprotonated. So there is going to be an equilibrium between these two forms. So let's move along further along the pH range, and now we are at a pH of 4.3, which also happens to be the pKa of the second carboxylic acid group. So now what's going to happen at the pH of 4.3 is exactly half of the carboxylic acid groups are going to be protonated and exactly half are going to be deprotonated. Okay, so as you went from 2.2 to 4.3, more and more of your glutamic acid molecules started existing in this form. Okay, because the equilibrium started shifting from this form to this form as you moved further along the pH. Now at a pH of 4.3, there is an equilibrium between this form and this form. Now if you were to move further along the pH range, we're at a pH of 9.7, which is the pKa of the amino group, you're going to have an equilibrium between the protonated form of amino group and the deprotonated form of amino group. As you move further along the pH scale, let's say at a pH of 12, 13, 14, the equilibrium is going to shift more and more towards this form right here. More and more of your amino groups are going to be deprotonated. Now let's take a look at what are the charges on each of these individual molecules. So here at a pH of 1, your glutamic acid is carrying a net charge of +1. So you have your carboxylic acid groups carrying a net charge of 0, amino group carrying a net charge of +1. So the net charge on glutamic acid is +1. The net charge on glutamic acid at this particular stage is 0. Okay, you have a +1 on the amino group, you have a -1 on the carboxylic acid group, you have a 0 on this carboxylic acid group. So the net charge on this molecule is 0. Now the net charge on glutamic acid at this particular stage is +1, -1, -1. The net charge is minus 1. At this particular point, your glutamic acid is carrying a net charge of 0, minus 1, minus 1, a net charge of minus 2. That brings us to at which point are all of the glutamic acid molecules carrying a net charge of 0. So your glutamic acid is carrying a net charge of 0 at this particular point, okay, which is above pH of 2.2 and below pH of 4.3. So at this point, pH of 2.2, there is an equilibrium between this form and this form. As you move along the pH range, the equilibrium starts shifting towards this point. So exactly where you have all of your glutamic acid molecules at a net charge of 0 is exactly between these two pKa's. So an isoelectric point is normally calculated by averaging out the two pKa values. But if you have amino acids that have 3 pKa values, which is the acidic and basic amino acids, the isoelectric point is calculated by averaging out the pKa's of the two groups that are really close to each other. So in this case, you have 2.2 and 4.3 that are really close to each other as compared to 9.7. So average of these two pKa values is going to be the isoelectric point of glutamic acid. Let's take a look at another example, this time of a basic amino acid, which is lysine. Okay, so lysine has carboxylic acid group and the amino group along the backbone, and it has an additional amino group as a part of the side chain, which is what makes it basic. So at a pH of 1, where the pH is less than pKa of all of the three functional groups, the lysine is in its protonated form. So it is protonated wherever it can be. So you have carboxylic acid group being protonated with a charge of 0. You have the amino group being protonated with a charge of plus 1. You have this amino group being protonated with a charge of plus 1. So the net charge on lysine at pH of 1 is plus 2. Now let's move along the pH range without taking any numbers. Let's just call it pK1. So what is going to happen at pK1, which is the pK of this carboxylic acid group, is this carboxylic acid group is going to start getting deprotonated. When the pH is equal to pK1, there's going to be an equilibrium between the protonated form of carboxylic acid and the deprotonated form. So at this particular point, where the pH is equal to pK1, your lysine is going to carry a net charge of minus 1, plus 1, plus 1. That makes it plus 1. Moving further along the pH range, now we are at a pH where the pH is equal to pK2, which happens to be the pKa of the amino group along the backbone of amino acid. So when pH is equal to pK2, exactly half of your amino groups are going to be protonated, exactly half of them are going to be deprotonated. So at this particular point, your molecule is going to carry a net charge of minus 1, plus 1, 0, net charge of 0. Moving further along, when pH is equal to pK3, the equilibrium is going to start getting shifted towards this particular stage, where pK3 is the pKa of the second amino group. So now this amino group is going to start getting deprotonated. So at pH is equal to pK3, there is going to be an equilibrium between molecules carrying net charge of 0 and molecules carrying a net charge of minus 1. So now at which particular point are you going to have all of the molecules of lysine carrying a net charge of 0? So remember, at pK2 there exists an equilibrium between lysine carrying a charge of plus 1 and lysine carrying a charge of 0. As you start moving along the pH, somewhere along the line, the equilibrium is going to start getting shifted towards this stage where the molecules are carrying net charge of 0. So as you go from pK2 to pK3, somewhere along the line in the middle, you're going to encounter a point where all of your lysine molecules are going to carry a net charge of 0. And that is exactly what the isoelectric point is. Again, going back to a simple definition of isoelectric point, especially for acidic and basic amino acids, it is simply the average of the two pK values which are really close to each other. So you have a pK value of around 2, you have a pK value of around 9, you have a pK value of around 11. So the isoelectric point for lysine as an approximation is 10. And that is it about amino acids.